This is Lecture 8, Development of the Nervous System. It corresponds with Chapter 8 of your textbook. You can skim the first section, Section 8.1, but please carefully read the rest of the chapter. Reminders and announcements. Exam 1 grades have been released. If you look through your exam and you see anything that doesn't make sense to you, or if you have questions about any of the material or any of the items, please don't hesitate to contact me. Your recommended readings this week are about facial recognition and people who are on opposite ends of the spectrum of facial recognition ability. So on one end is people who are very bad at facial recognition, and this is people with prosopagnosia, which is also called face blindness. And you can watch this video from Oliver Sacks or read the story about a person who suffers from prosopagnosia. On the other end of the spectrum are people who are very good at recognizing faces, super recognizers. And there's an article about how this ability is used in a police force in London. They recruit people specifically who are very good at recognizing faces so that they can see if they can identify people from like CCTV video who have committed crimes. And then there's a test you can take that was developed by a research group, I believe also in the UK, to see if you might be a super recognizer, which is considered to be about 1% of the population, but in general you can see how good you are at facial recognition.
Alright, so jumping into Chapter 8, Section 8.1, I'm not really going to be covering. I recommend skimming it, but I'm not going to be taking any homework information from it or exam material from it. They do mention that genetics should be conceptualized like a blueprint. This is a concept we had talked about before, but I just want to bring it up as a reminder that genetics are not a complete determinant. They are more just like a blueprint, like when you're making a house and general instructions, but there are other factors that influence the final product. So Section 8.2, the neurobiology of development. In vertebrates, embryos look very similar, both in the body and early brain development across species. So you see this figure down here. There's the embryos of a salamander, a chick, and a human, and you see that they look very similar. So early brain development, very early brain development in utero looks very similar across vertebrates. So a rough timeline for the gross development of the human nervous system. During weeks 1 to 2, so the first 14 days, day 1, of course, the sperm joins the egg, and this is fertilization. This creates a single cell human, which is called the zygote, and these cells divide, starting on day 2. Continue division until around day 15, when they form this flat plate called the embryonic disc, flat plate with a little raise in the middle. Weeks 2 to 8, we have an embryo, this sheet of cells with the raised middle. Week 3, the neural plate, which is this raised bit in the middle, folds and curves to form the neural tube. And the neural tube is extremely important structure from which the brain and the spinal cord develop. So it's literally just this sheet of tissue that folds and curls around to make this little tube. And from that tube, all of the cells are dividing and being created, and this will eventually be the brain and the spinal cord. Week 7, the embryo starts to look like a little person. Week 11, the brain starts to look humanoid, so it starts to look like that human brain that we know. And then by the 9th month, the brain starts to look like an adult human brain, but the cellular structure is a bit different. So the origins of neurons and glia. The neural tube is the origin of all brain cells, as I mentioned before. Neural stem cells are cells that can self-renew. So neural stem cells can create many different types of cells. In fact, all of the cells in the central nervous system are created from neural stem cells, and this includes both neurons and glial cells. For adult humans, our ventricles, which if you remember are those spaces in the brain filled with cerebral spinal fluid, are lined with neural stem cells. And this area is called the subventricular zone. Neural stem cells create progenitor cells, and which in turn create neuroblasts and glioblasts, which are the precursors to neurons and glial cells. And neuroblasts and glioblasts can create all the different types of neurons and all the different types of glial cells. So cells differentiate, they turn into the different types of neurons and glial cells based on what genes are expressed or turned on. Cells that are turned on will make, or genes that are turned on will make specific proteins, which produces this differentiation in the cells. One process by which this happens is gene methylation. So there's a compound called methyl groups, and they bind to and suppress a base pair in DNA, in genes. And this is one way in which genes are expressed. So some genes are turned off, which expresses other genes. This is also one of the main mechanisms of epigenetics, or passing on characteristics that are gained in life, passing those on to offspring. Cell differentiation is regulated by a series of chemical signals, and these are called neurotropic factors. So neuronal growth and development, after neuroblasts and glioblasts are formed, they migrate to the correct area of the brain. So some cells will migrate to the frontal lobe or frontal area. Some will migrate back to the visual cortex area, at which point they differentiate and grow dendrites and axons. Neurogenesis is mostly finished at about 5 months after conception. Except in the hippocampus. In the hippocampus, new neurons are actually developed throughout the entire lifespan. So it's kind of a special part of the brain. If you remember, the hippocampus is responsible for a lot of the memory functions in the brain. So you can see why we would need new neurons in that area of the brain to help form new memories. And the creation of new glial cells continues throughout the life. So unlike neurons, where not many new neurons are being produced throughout adult human life, new glial cells are continuing to be formed throughout adult human life. Cell migration starts shortly after neurogenesis, so shortly after those neuroblasts and glioblasts are created. Cell migration takes about 6 weeks, and after which time differentiation happens. The cells are differentiated into different types of neurons. After the cells are differentiated and become the specific types of neurons, they then mature, which means that they form dendrites and axons, synapses between the neurons. And neuronal maturation can continue through adulthood in certain areas of the brain. Also remember that the cortex is organized in layers. So you remember there were 6 layers in many areas of the cortex. The subventricular zone functions as a sort of map that sends cells to different areas of the brain. So if a new neuron is in a certain part of the subventricular zone, it's going to be sent to kind of a corresponding part of the brain. Radial glial cells are special glial cells that create kind of a pathway, sort of create a bit of a rope that neurons follow to the correct layer of the cortex. Some cortical neurons do follow chemical signals instead of these radial glial cells, but most of the migration happens through radial glial cells. And cortical layers develop from the inside out. So it's just like building a house. You would build the foundation first, and then the walls, and then the roof. The cortical layers are built the same way. So for neuronal maturation, first there's dendrite development, and this involves dendritic arborization, arbor being like tree. So this just means like the branching of the dendrites. And then spine growth, or these little nubs that are created on the dendritic branches. And then there's axonal development, which compared to dendritic development is very fast. Axons, if you remember, can go really far, very far through the body. So there are some axons that go all the way from the central nervous system down to your big toe in your foot. Axons take cues from the environment, from the body, to reach the target. Simple synapses form in the brain 5 months after conception, and synapses start to increase very quickly after birth. So after the baby has been born, they're suddenly exposed to this whole world of sensory stimuli and experiences that they didn't have inside the mother's womb. So they're quickly forming synapses to account for and learn from these new experiences. On the flip side, there's synaptic pruning and cell death. So within the nervous system, there's kind of a principle of use it or lose it, meaning if synapses are not being used or neurons are not being used, they get removed. And this is one way in which the brain maintains efficiency, so it doesn't have wasted connections or wasted cells. Synaptic pruning is, of course, elimination of these synapses that are no longer needed. There's a massive period of pruning that happens between early childhood and puberty, and this accounts perhaps for some of the rapid behavioral changes you might see during this period, and it also accounts for some of the critical and sensitive periods that we're going to talk about a little bit later. Critical periods are this time during which certain functions such as language are particularly easy. So for kids, you might have heard that kids pick up on language more easily than adults, and this is absolutely true. It's in part because their brain is literally physically ready for language development during these times. And actually, if kids are not exposed to language until later in their life, they may not be able to gain full fluency in language abilities. And we'll talk a little bit more about this later. Cell death or apoptosis is happens when neurons are deprived of neurotrophic factor, and this triggers programmed cell death. And this also happens when neurons are no longer needed. So frontal lobe development is rather special. The frontal lobe is the last area of the brain to fully mature, and it continues to develop after 20 years. We're even finding that it might develop into the early 30s. There's one area of the brain, of the frontal cortex, called the dorsolateral prefrontal cortex that develops particularly late. So you can see on this bottom right figure that the red denotes parts of the brain that are less fully mature throughout different time periods. So we have a 5-year-old, preteen, teen, 20-year-old. And so dorsolateral prefrontal cortex is rather immature in a 5-year-old, even in a preteen. It goes more toward green, which is like partially mature as we get to teenage brains. And then even a 20-year-old, it's still this green, light blue color, indicating that it's not fully mature. While most of the rest of the brain is this darker blue and purple color, indicating that it is fully matured. So the dorsolateral prefrontal cortex is responsible for or at least takes part in a number of functions such as working memory, cognitive flexibility, planning, motivation, and attention. Synaptic pruning in this part of the brain continues after 20 years, likely until early 20s in many people. It happens a little bit later in males compared to females. And you can compare the behavior, just that you've observed, of individuals in their early 20s compared to individuals maybe in their early 30s on some of these dorsolateral prefrontal cortex functions. So planning, motivation, attention, things like this, you might see a little bit less maturity in some of these functions in people in their late teens as compared to people in their late 20s. Okay, glial development. So astrocytes and oligodendrocytes start to develop after neurogenesis is finished. And if you remember from our previous chapters, that these two types of glial cells are responsible for the myelination of axons. Myelination is a sort of considered to be a crude measure of maturation in the central nervous system because it is one of the last parts of brain development. And different areas of the brain are myelinated at different times. It's been observed that the first places to be myelinated are sort of the basic motor and sensory areas that you need first in development. So when you're young, it makes sense for survival to have at least the basic motor and sensory areas before some of these higher cognitive areas develop. Okay, so moving on to section 8.3, inferring brain maturation from behavior. There's a number of different types of behavior that we can connect the sort of behavioral and cognitive development of kids onto what we know about the brain developing during that time. So motor behavior is one of the first things that you observe in babies. There are all these milestones that parents are often obsessed with. There's something that we observe called motor babbling, which is random or spontaneous hand and finger movements that actually consist in sort of snippets of movements that the baby's going to later use in skilled fine motor control. And it's believed to be a way to practice hand movements. So they kind of practice these little hand movements, but in a non-purposeful way. And this typically happens between months 1 to 3. By 4 months, babies can typically grasp objects in a more goal-oriented manner, although rather crudely and not in a very dexterous manner. By 10 months, they can use the famous pincer grip with their thumb and their index finger. So if you see parents giving their babies Cheerios or putting just like loose peas or something like that on their plate, the parents might be helping their kid practice their pincer grip, which is this ability to grab an object with the index finger and the thumb. And that's considered a pretty significant motor milestone. Increased fine motor control, so like the pincer grip, corresponds with the myelination of the motor cortex axons. So before I mentioned that myelination is kind of the last thing that happens in terms of maturation of or the gross maturation of a part of the brain. And so once an area is myelinated, the kid is able to do that activity to some degree. Interestingly, the increase in fine motor control is actually associated with the thinning of cortical areas that are associated with motor control. This might seem confusing because we've talked in the past about thickening of the cortex being associated with better functioning in those areas. The thicker the cortex, often the higher intelligence, things like this. But thinning is actually sometimes associated with increased efficiency of neuronal processes in young brains, possibly in part to synaptic pruning that has created increased efficiency of the connections between neurons. And also myelination increases the white matter, which decreases by proportion the gray matter. Okay, so language development. Children reach language milestones in a pretty fixed order, and this is across all different languages and cultures. At around 10 months, most kids are babbling. So this is like motor babbling, but this is with language. So in language, babbling is speech sounds that are contained in that native language, but are not real words. And certainly everyone has heard this. Kids just kind of speaking nonsense, babies saying syllables, da, 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 ba, 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 things like this. They're actually practicing the building blocks of language. And this will turn into their developing the motor skills and also trying to connect what they hear to what they're producing. So babbling is extremely important in kids. At around 12 months, they might be saying a couple words, 5 to 10 words. And there's a large increase in neuronal connectivity in Broca's area, which is this speech motor area between 15 to 24 months. Around 12 months, the vocabulary has risen to 200 to 300 words quite considerably. Cell division and migration is happening in language areas. It's mostly complete in language areas. There's increased connectivity and axonal myelination in those areas as well. Around 3 years, the vocab is rather large, 1,000 words. They're speaking in simple sentences. They can communicate rather effectively for the most part. Language acquisition is mostly complete by age 12. Of course, vocabulary is going to increase, etc., but they're speaking mostly like a basic adult. And notably, muscles that are required for coordinated speech develop before speech emerges. So babbling is probably part of the reason for that. They're practicing these muscles and these motor actions that are needed for speech. Notably, not all kids meet these rough milestones at the same time, so language is sometimes rather delayed in kids who later turn out to have perfectly normal language abilities. It's not always clear why that is, but that can be rather stressful for parents, and it's just worth noting that it doesn't always mean the kid is going to have autism or some other kind of language issue. It's just sometimes brains develop differently in kids. So language is associated with, or increased language processing is associated with a thickening, in this case, of the left inferior frontal cortex. So this is around that Broca's area that we were talking about before. There is a critical period for language. So again, a critical period is a time during which a skill must be developed, or it may not be able to, after this critical period, it may not be possible for an individual to develop that skill, at least at the level of their cohorts. So roughly between 5 years and puberty, it's believed that a kid must be exposed to language and at least start to learn language, or they may never develop fully fluent language. There's a very famous case that you might have seen in an earlier psychology class if you've taken other psychology classes. This girl, Jeannie, who was very severely neglected and abused and was not exposed to language early in her life. She was kind of brought in by these researchers, and they tried to teach her language, but she never was able to fully develop language. And if you'd like, there's a link to a really quick documentary on this girl. Notably, this is, of course, a case study. There have been other case studies that have found similar effects. It's obviously something that we can't do experimental work on. It would be extremely unethical to deprive kids of language and see what happens, but there have been enough individual cases of this that it does seem to be ring true that kids must be exposed to language previous to puberty. In terms of problem-solving abilities, people may have heard of Piaget, who had stages of cognitive development. I'm not going to go into the individual stages very much. That's more of a developmental psychology, more of what you would learn in developmental psychology class. But roughly there's these 4 general stages: sensory motor, pre-operational, concrete operational, and formal operational. And Piaget believed that kids sort of had these spurts of cognitive development. So they would be able to do a certain amount of things in this stage and couldn't do any of the skills from later in the stage. And this has been a little bit not everyone completely agrees with all the stages and everything that Piaget said, but it does map on fairly well to what we know about growth spurts in the brain. So during these approximate typical age ranges of the stages, there are also these growth spurts in the brain, and in both the body and in the brain with increased neurons. So growth spurts, probably familiar with this term, they're sporadic and finite periods of sudden growth. This is goes along with the principle that the brain and body do not grow uniformly. It's not a nice, even growth line. It's comes and goes in spurts. So roughly 3 to 10 months, there's kind of a growth spurt, and then growth might level off for a little while. 2 to 4 years, you see another spurt of growth that might level off, so etc. for 6 to 10, 10 to 12, and 14 to 16. During these times, brain weight can increase by as much as 5 to 10%, which is quite a large amount. And this brain weight is accounted for by increase in synapses, neurons getting bigger, new astrocytes being created to support the neuronal growth, and then increases in the myelination. So one way that we can observe this sort of these phases or stages of development is that children don't always seem to learn the simplest tasks first. Sometimes they seem to have gained more complex skills at an earlier age. And this might be in part to different brain areas developing, not the simplest ones first, for example. So there are these three tasks that these experimenters did. The first was a displacement task, where food is earned when a specific object is displaced or moved. So in the picture, you can see they did this task with both monkeys and with kids. I'll talk about the one with the kids. So if they displace the little purple pyramid, then they get a food reward. The next is the non-matching to sample task, where a novel object can be displaced for a reward. So regardless of what the new object is, they're shown two objects, and regardless of what the new object is, if that one is moved or displaced, then they always get rewarded for that one. So the child learns that just always move the new object and you get the reward. They can do this task around 18 months, and it's believed to be dependent on temporal lobe functioning. So the next task is the concurrent discrimination learning task, and this is like a trial and error learning task where the kid discovers that certain objects are always associated with reward. So it's not that novel objects are associated with reward, it's that, for example, within pair 1, on day 1, day 2, etc., the green box is always going to be associated with the food reward, and then on the second pair, this yellow L is always going to be associated with the food reward, etc. And this task is believed to be dependent on basal ganglia functioning. And children can do this task around 12 months, which is surprising because it seems like a more difficult task. The child has to remember which objects are always associated with the food reward. It seems like it would be easier that the kid just has to remember to displace the new object, as in the non-matching to sample task. And it's believed that kids can do this kind of more complex concurrent discrimination learning task at an earlier age because basal ganglia functioning might develop, the basal ganglia as a structure might develop more quickly than the temporal lobe, making it possible to do this more challenging task before the seemingly easier task. Okay, so 8.4, brain development and the environment. Early life experiences shape brain development. There's a loss of plasticity in the brain in adulthood, but there is still some plasticity. But of course, early brains are very plastic and are constantly changing based on these life experiences. When one brain area is damaged, the function of that area might be taken over by other brain areas, and this is especially likely to happen when a young brain is damaged. So for example, when the Broca's area, which again is this area in the frontal lobe that's associated with language production, if Broca's area is damaged, a patient may regain some of the language function when other adjacent cortical areas start to take over the function of Broca's area. So they might go through a period of not being able to produce language fluently. However, over time, they may regain some of this function due to adjacent cortical areas taking over that function. More experiences and a more stimulus-rich environment are associated with higher intelligence in general. Experimentally, we see that rats with a richer early life environment are better at maneuvering through a maze. So I think the book gave you an example of a scientist keeping some rats in the lab, which was a pretty unstimulating environment, and then keeping some rats in his kitchen, which was stimulating because his wife was chasing them with a broom, etc. And they were just getting more exposure to different experiences and stimuli. Observational, there's a very famous and tragic study of children raised in Romanian orphanages in the 1970s. So there was a rather tyrannical communist rule in Romania in the 1970s, and I think birth control and abortion were illegal, and there were a lot of children in orphanages and not a lot of money to take care of them. So these children in these Romanian orphanages were raised, many of them in one crib. They weren't being picked up, they weren't being held. So it was extreme negligence, essentially. And scientists found that these brains of human children in extreme neglect are smaller. And the children have a lower IQ overall later, and in general just don't fare as well as children who have a more rich early life environment. So you can see over here to the right, this is, I don't think it's necessarily from the Romanian children's study, but this is an example of a brain of a normal 3-year-old child, and then the brain of a 3-year-old child who was raised in extreme neglect, and you can see how small, how much smaller it is compared to the 3-year-old child, and you can imagine how that will follow that child throughout their life. In addition, we see different neuronal architecture in rats who have more stimulating environments during their development, more dendritic branching, more connections between, more synapses and connections between neurons. And we also know that specifically tactile stimulation is very important. So there are a number of studies where baby rats that were raised in a lab were like brushed. They literally used like a paintbrush to sort of pet the baby rats to give them sort of a standardized form of affection, I guess, or tactile stimulation. And both motor and cognitive functioning was improved in the rats who were brushed as compared to the rats who were not brushed during development. And this speaks to the importance of holding babies. So going back to the children in the Romanian orphanages, those kids were not being held. There weren't enough resources, people didn't have time, or perhaps didn't understand the importance of just holding the babies once in a while. And that could have had a really large impact on their later functioning. So for our music lovers, it does appear that early exposure to music affects neurons in the auditory system. There's this particular study where there were structural changes after just 15 months of musical training in children, where there was, there's increased gray matter. If you look down at the pictures on the right, on the left, increased gray matter in the primary motor area for people who played instruments versus controls, increased density of the corpus callosum, so that would be white matter. If you remember, that's the connections between the two lobes, so increase white matter in the tracts that connect the two lobes, and then increases in the primary auditory area as well. And this was anatomical MRI as a method, which shows differences in the amount of gray matter, in this case differences in the white matter as well. Early exposure to music appears to affect neurons in the auditory system as well, so connections between neurons. This particular study, Steele et al. 2013, shows diffusion tensor imaging, which is a method that studies the connectivity between brain areas, or the white matter tracts between areas, rather than just looking at the volume of gray and white matter. It's looking at the strength of connections, how many white matter tracts are connecting these brain areas. This study suggested that there might be a sensitive period from ages 6 to 8 in which music can be particularly influential on the development of motor skills. And so they found that kids they looked at the brains of musicians, and individuals who were learning during the ages of 6 to 8 had particularly strong connections in these areas of their brain. So looking back, I think we're looking at the same areas actually as the previous study, so they're not labeled, but this is the motor area here. You can see there are strong connections, very strong connections. The red and the yellow indicate strong connections in the motor area of these individuals who learned music ages 6 to 8. Again in the corpus callosum in the second picture, and then again in the temporal lobe, the auditory cortex as well. Sensitive period is a little bit different than critical period. The idea of a sensitive period is that the brain is most primed to acquire a skill during this time. It doesn't mean you'll never acquire the skill if you learn outside of that time, so it's not as strong as a critical period, but it's still a time during which the brain is particularly receptive to learning a specific new skill. So there are prenatal events that can affect brain development. In fact, most prenatal events can affect brain development. Fetuses can actually hear sounds at 30 weeks and can recognize a mother's voice in utero soon after they start to recognize sound. So there's been a number of studies about this, but there's this one particular study where researchers had a baby listen to their mother's voice reading a poem compared to another woman's voice reading a poem at the same volume. And the baby's heart rate quickened when they heard the mother's voice reading a poem. So they actually had a recording of the mother's voice. The mother wasn't reading it out loud. And this, along with a number of other studies, make a pretty compelling argument that the baby can recognize its mother's voice, can make a differentiation between another person and the mother's voice, even while still in the womb. Unfortunately, prenatal events can have a very negative effect on development in a baby. So teratogens are any agent that causes abnormal development due to prenatal exposure. This includes maternal stress. If a mother is under a huge amount of stress, large amount of stress, prenatally, this can affect the baby's development. Drugs, alcohol, tobacco, cocaine, for example, can have a big effect on development. Can have pretty profound and devastating effects on development of a baby. Lack of oxygen for a period of time can negatively affect development. And the degree to which teratogens affect the development of a baby depends on when they're exposed. So different functions, different parts of the brain and the body are being developed at different times prenatally, and based on when the most development is happening for a given part of the body, so with the hands, for example, or the face, teratogens can have a more devastating effect during periods of faster growth, for example. So fetuses that are exposed to teratogens during development of the midline of the face, which is like the nose and the eyes, it's rather rare, but they can actually get cyclopia, which is comes from cyclops. So they can be born with one eye in the middle of their head. This is actually a really important time for brain development too, so typically these babies unfortunately do not survive. This would result in a miscarriage. But sometimes animal babies survive that have cyclopia. And I spared you the pictures on the slide, but if you want to look online, you can type in like cyclopia kitten, or I think maybe like baby goats are often tend to be born with cyclopia. So you can see the kind of the devastating effects. Interestingly, the same teratogens that can cause cyclopia, because they affect the development of the midline of the face, they can also cause two-headedness. So exposure to teratogens during the same time can cause kind of the opposite effect, which is to create to split the head in general and create two different heads. And so again, I've spared you pictures of this on the slide, but you can, this has happened in kittens and other animals. And it has happened in people too. Although I'm not sure having two heads, I'm not sure if people would survive. There are, like twins that are connected such that it looks like they have two heads, but I don't know if one person who has two heads would necessarily survive. More minor issues that can happen due to exposure to teratogens are minor physical anomalies, and these are small physical differences, mostly in the hands and the face. Also happens in the feet, that indicate abnormal development during specific months. So if you look to the right, there are minor physical anomalies that may happen from exposure to toxins during the first trimester. And this includes like kind of a tongue that has like big lumps or is really rough. You see the hand there, there's like they call it like a single transverse palmar crease. So really, really strong single crease in the hand. Ears that look malformed. This in the eyes, they may have eyes for a Caucasian baby, like eyes that are not that cover up the tear duct, where you can't see the tear duct, and then feet where the big toe is separated from the second toe quite a bit, so like an increase in the toe gap between the first and the second toe. Notably, this, like if somebody has these physical features, it doesn't always come from teratogen exposure in the first trimester. These are just, it's kind of a syndrome, a series of physical anomalies that you might see if somebody has exposure to poisonous or developmental abnormalities in this first trimester. These minor physical anomalies are associated with a number of psychological disorders, including autism, schizophrenia, and Tourette syndrome. And the idea is that because we see these increased physical abnormalities that are associated with teratogen exposure in the first trimester, because we see more of those in people with these psychological disorders, there's probably something that's happening in the brain too when people are exposed to teratogens in this first trimester that's making them more likely to have these disorders. So it's highlights the possibility that this might have happened to somebody with these abnormalities. Okay, hormones and brain development. Of course, the sex of the baby is mostly determined by genetics. There's the XX chromosome that would normally create the female sex. XY typically creates male. As many of you know, there can be situations of XY females and a number of different chromosomal abnormalities can happen that result in male or female sex or something in between. Interestingly, the female brain is the default. The male brain is sort of the female brain that's exposed to testosterone. So the Y chromosome steps in during development and creates gonadal tissue, or develops the gonadal tissue into testes. Testes in turn secrete testosterone, which leads to the development of male reproductive organs. And then testosterone also affects the brain and creates some differences between male-female brain, as such as where the neurons grow, when they die, synaptic activity. A lot of basic processes can be different in the male versus female brain. If you look up on the image here on the right, the orange areas are areas that are generally larger or thicker cortex in females versus the green areas, which are generally larger in males. And again, these are broad generalizations. It doesn't mean it's the same for every male and every female. Okay, gut bacteria and brain development. So we've talked previously about the enteric nervous system, or the ENS. And part of this, a very important part of this, is the microbiome, which is gut bacteria. The microbiome is inherited from the mother, typically during vaginal birth and skin contact. So in the case of C-sections, where the baby doesn't pass through the vaginal canal, there's some people believe, and some research backs this up, that babies are not inheriting as much of the microbiome if they're delivered during C-section. And this could lead to problems later on, both problems within the enteric nervous system and within the nervous system in general. There's a risk of some disorders that's associated with abnormal development of the microbiome. So, for example, autism the microbiome in autism has received a lot of attention, and there seems to be some indication that abnormalities in gut bacteria may in some cases be a risk factor for developing autism. And there have been experimental demonstrations as well of associations between behavior and microbiome, where people with behavioral disturbances are given kind of an infusion to improve their microbiome, and behavioral problems have improved. And so this is a lot of this research is still in the very early stages, but it's very interesting and from my perspective, very promising in terms of developments that may happen in the near future. Injury in brain development, there's sort of a time during which injury can be most devastating. So the worst time is in the last half of pregnancy and the first months afterwards. So if you've ever heard of like someone dropping a baby on their head, especially a newborn infant, this really can be very devastating. I mean, it can be bad for any kid, but especially devastating for a new infant. In later parts of development, plasticity of the young brain can make it more resistant to injury. Drugs, many prescription drugs, even common prescription drugs, can be harmful to a fetus, harmful to development. The FDA in the US has classes of drugs for pregnancy indicating whether women should, giving women some information about whether or not they should be taking these drugs during pregnancy. And so a case study of the effect that medications can have on brain development, there's the very tragic case of thalidomide. This was a drug that was marketed in the 1950s for a number of things, but in Canada and Europe it was marketed as a cure for morning sickness. In the US there were some cases, but I think it was never fully approved by the FDA, so it wasn't as devastating in the US. At the time, many scientists believed that drugs couldn't cross the placenta, so not as much attention was paid to medications that women were using during pregnancy. Unfortunately, it turns out that thalidomide is a teratogen, and it caused at least 2,000 infant deaths, 10,000 birth defects, I think 5,000 of which eventually died soon after. And the birth defects were rather severe, so they caused serious malformation of the limbs, sometimes just a lack of formation of the limbs at all, deafness, blindness, cognitive problems, just a whole range of issues. You can see on the bottom right, here's a little girl who was, they called them thalidomide babies that have been affected by this. And she didn't develop arms at all, has adapted quite well to playing with toys with her feet. So interestingly, damage was very dependent on the time of exposure. So if thalidomide was taken by the mother around 20 days after conception, there was central nervous system damage. If it was taken 21 days after, eye damage, 22, ears and face damage. And you can imagine that people were taking it multiple days, so multiple things could be affected. And of course, there was variation across individuals. If it was taken after 42 days, there was minimal to no damage. So it was if it was taken after this certain period in development, it didn't affect the babies as much. And as tragic as thalidomide was, it did bring attention to the effects of drugs on fetal development, and people have been much more careful to make sure that medications have been tested before they're okayed to be used on pregnant women. So other sources of abnormal brain development, there are internal sources, things that just happen kind of naturally. One of these cases is spinal bifida. Spinal bifida is when the spinal tubes do not close completely, and a lot of times there's a abnormal formation of the spinal column in general. It can look, it's pretty sad looking. There's usually just kind of like a hole in the back of the spine. Again, you can look it up, but I spared you the picture. It primarily comes from non-genetic causes, and this is one reason that pregnant women are advised to take folic acid, which is a B vitamin. As well, many grain products, such as cereal and bread in the US, are fortified with folic acid in large part for this reason. Folic acid plays a role in cell division, so normalizing cell division throughout development can help prevent spinal bifida. And scientists estimate that a large amount of spinal bifida cases can be avoided just through supplements of folic acid during pregnancy. Effects of abnormal brain development can be subtle, with the full effects emerging later in life. So schizophrenia is a really good example of this. Schizophrenia is considered by most to be a neurodevelopmental disease, meaning that it's a disease that is caused by abnormal development of the brain. And this abnormal development typically begins, at least in part, in utero, although the symptoms don't emerge until much later. So schizophrenia is considered to be about 50% environmental factors, 50% genetic on average. And most genes that are linked to schizophrenia code for brain development, particularly synaptic plasticity, transmission, and functioning of the frontal lobes and hippocampal regions. Typically, children don't show huge changes that would indicate that they're going to develop schizophrenia later. It's usually more subtle, behavioral and motor and emotional issues. So kids might have depression, they may suffer from attentional problems, or a lot of times just kind of be like a little bit unusual, maybe socially awkward, and just a little bit different. And then in their mid to late teens, they may start to develop some of the psychosis symptoms, and typically it comes out completely in late teens, early adulthood, with it happening just a little bit later in women, where like early 20s to mid 20s is more typical for women. And this is likely in part due to the later stages of synaptic pruning and the frontal lobe development happening later in life. So as we talked about before, frontal lobe doesn't continues development into the 20s, and schizophrenia, frontal lobe is implicated in the pathogenesis of schizophrenia, and so the later development of the frontal lobe is probably one of the main reasons that schizophrenia emerges so much later in life, even though many of the things that happen that create a risk for schizophrenia happen early in life. Developmental disabilities, obviously there's a range of developmental disabilities, but in general, for developmental disabilities, you see less dendritic growth and fewer spines. So there can be a range of causes. This can be birth trauma, so oxygen deprivation during birth, which can lead to a cerebral palsy. It can be malnutrition, mother was not getting adequate nutrition during pregnancy, or baby is not getting it during early years. Drugs, as we talked about before, environmental abnormalities, so like the sensory deprivation, like the tragic case of the kids in Romania. Genetic abnormalities, of course, such as Down syndrome, where they have the extra chromosome. And then prenatal disease, if the mother gets an infection, such as rubella, which is like the German measles. Rubella has been linked to, a woman getting rubella during a pregnancy has been linked to birth defects later on, and developmental disability.